Now moving on to our next speaker, Professor Gautam Menon from Ashoka University. He will be talking about activity, phase separation, and the nucleus. Over to you, Professor Menon.
Okay, so it's wonderful to be here at this meeting, especially to see Deepak and Musansir again. And like most people here, I owe them a debt of gratitude. My own interests have been somewhat different from sort of exact stat mech for a while. They've been more concentrating on the biophysical range of things. And a question we've been thinking about for the last at least 10 years is what determines the large-scale architecture, the nucleus of human cells. And this is an ancient problem. It's more than 100 years old. And the interesting thing is that it's not a problem in biology. It needs both physics and biology for reasons that I will explain as we go along. We have some answers to this question. And the answer is that you need a combination of activity, confinement, looping, which may be less important, the looping of chromosomes. And the last point is something called phase separation in this context, which I will explain as we go along. And so let me just remind you of some bare facts that you might need to remember. The other is that, you're made up of cells. All of us are made up of cells. Each cell has a nucleus. There are 23 pairs of chromosomes in the nucleus, and each chromosome is a single long DNA molecule. So there's the stuff inside this little bag, the nucleus itself, and the chromosomes are labeled 1, 2, 3, all the way up to 22, and then there's an X chromosome and a Y chromosome that can enter. So there's a distinction between what we thought prior to about 1990, 1992, and earlier ideas regarding the organization of chromosomes inside the nucleus was that these are just long, each is a long polymer, there are 23 pairs of these. This probably looks like spaghetti or noodles or Maggi noodles or something. And of course, the term that is used to describe chromosomes in their natural habitat, so to say, is a word called chromatin, which accounts for the fact that this just isn't DNA, but DNA with proteins attached to it, little pieces of RNA, etc., floating around. So this whole complex material is together called chromatin. So the question is, how is chromatin organized inside the nucleus? And zeroing in on specific chromosomes, how are specific chromosomes organized inside the nucleus? So post around 1992, our whole conception of what was going on inside the nucleus changed. It was realized that chromosomes are actually territorial. They occupy fairly well-defined regions on their own within the nucleus. And these arrangements of the chromosomes are not random arrangements. So we will be talking about human cells in the space in between division, because cells, when they divide, do far more complicated things. So this is about the 80% of the time cells spend between divisions. And that's the time in the organization that I'm describing will set in. The most important thing to remember here is that there's a lot of what are called active processes, that non-equilibrium processes that happen on chromatin. Chromatin is not in thermal equilibrium. But then the question is, how do you describe such a system that is not in equilibrium? And how do you extract anything that's useful in terms of either insight or in terms of being able to compare to a real experiment, to experimental data? So the physical context here is that this is an active system. An active system has input of energy at the microscopic scale, the nanoscale in this particular system here. And that really occurs through the consumption of a small molecule called ATP. So you have a lot of ATP consumption that happens, and this happens because chromatin is constantly being remodeled, being opened up. There are machines, various machinery, the polymerases that walk along the DNA and make RNA molecules, etc. And all of this contributes to the fact that this is an extremely non-equilibrium system, and activity is at the center of all of this. So we can start with a hypothesis that is activity that is not the same across gene-rich and gene-poor regions that really determines large-scale nuclear architecture. And that's a good starting point to think about these ideas. And I'll tell you how we make it more quantitative. So the way we make it more quantitative is to imagine bunching up or grouping all of the DNA, which really is defined at the level of base pairs, chromatin, into different regions depending upon the number of genes that they contain. And more active regions where there's more consumption of ATP, are associated with regions that contain genes that are being more actively written out. And we say that there are larger mechanical fluctuations in those regions, which can be modeled once you coarse grain sensibly enough as a higher effective temperature. There are also less active regions which see smaller fluctuations, and the background here is physiological temperatures. And this is a low effective temperature compared to the high effective temperature that comes in really from non-equilibrium fluctuations. So take a simple model, look at the gene density, because we know the human genome, we know where the genes are, look at the gene density across each segment. Just take the top 5% of these monomers by gene density, call them active, and set a larger effective temperature for them. The nice thing here is that this really makes connections with the biology, because you have to know where the genes are. And so each little chromosome carries a fingerprint with it of what are the active regions, what are the inactive regions, what's being written out, what's not being written out. And therefore chromosome 18 and 19 have very different levels of activity, and chromosomes as from 1, 2, 3, etc., etc. And so the next step, of course, is to simulate individual chromosomes within the nucleus, change the nuclear shape. This is a spherical nucleus, a flattened nucleus, etc. And look, for example, at specific chromosomes. Where is chromosome 18? Where is chromosome 19? And you can look at experimental data, which looks at chromosome 18 and 19, and chromosome 12 and 20, any chromosome for which there is enough data. And chromosome 18 and 19 have the same length, roughly, but very different genes per unit length. And chromosome 12 and 20 have different lengths, but the same number of genes per unit length. And therefore, you can now take the results of the simulations and compare it. And so that's how it does. And you can see that it does fairly well. It shows that 18 and 19 should be fairly separated in terms of where they are relative to the nuclear center, whereas chromosome 12 and 20 should be fairly close in terms of where they are relative to the nucleus. These are distribution functions. I'm not going to define this because I don't have enough time to go into this. Here is the picture, the original pictures that actually showed you what the formation of chromosome territories, the fact that chromosomes didn't overlap all over the place like a dish of noodles or pasta. And you can recover this in these simulations as well, whichever initial condition you start out from. Each chromosome here is colored a different color. And there's a natural way in which chromosome territoriality follows from the model here, and it's fairly robust. Look at the center of mass distribution. So here is each chromosome 21, 22, 19, 28. And there's a rough behavior according to chromosome size here. The larger chromosomes tend to be outside. The smaller chromosomes tend to be inside. You can compare this with the predictions of the theory. And that's how it does. And it's actually not too bad if you think about it. So these predictions agree fairly well with data, even at a quantitative level, and they agree with our broad understanding of not just one set of measurements, but of many different measurements, which is fairly encouraging. And it's usually not easy to get any reasonable agreement between theoretical model and experiment in biophysics of this sort. So that should make us a little bit happy. The second part of the talk is about new paradigms in thinking about how the nucleus is organized. And the paradigm here that you pick up any issue of PNAS or Biophysical Journal, etc., is the issue of liquid-liquid phase separation. And the idea here is that much of the organization of the cell's cytoplasm, the organization of the cell's nucleus, happens because it really can be thought of as liquids with different levels of miscibility in each other, and droplets of one inside the other. And maybe it's in these droplets that you can enrich specific biochemical components so that they're there and you can be used at higher density than they would be if you just distributed them uniformly. So this is useful. Now, these questions really came up in the context of something within the nucleus, which is called the nucleolus. And that's a picture of the nucleolus. It's the largest sort of sub-organelle within the nucleolus. It's the most prominent of the nuclear bodies. And what it does is to assemble the cell's ribosomal RNA that's needed for ribosomes, which make proteins, which are present outside. So you make the RNAs that go into the ribosome, you export them from outside the nucleus to the nuclear pores, you take them and you construct outside this whole machinery for which a Nobel Prize was awarded some years ago, including to Venki Ramakrishna, the structure of the ribosome. And this little structure here is dense, it's very visible. And the question is, how is it made? And the ideas here have coalesced around the fact that the hypothesis that this is really a multiphase liquid condensate. And so then, you can, here's a set of examples where you look at water, vegetable oil, silicone oil, all of which have different levels of surface tension. And then you can look at how they form little droplets within other little droplets in terms of where these numbers are. You can in vitro reconstitute elements of what goes into the cell's nucleus and ask questions about those and how they organize. And then you can look at the real sort of the cell example here, which is from Xenopus laevis, the oocytes of Xenopus laevis, where you really see how regions that define the nucleolus begin to look like the little phase-separated droplets that you have to your left as they begin to come together, and then they begin to assemble and they begin to coarsen. So this here is really at the level of comparing different pictures to each other, but you can make this much more precise and much more targeted, and actually show that you can adding or removing components or restructuring the components that enter the biological system enables you to recapitulate or either take away or add to the level of phase separation that you see in the soft matter system to your left. So the nucleolus is kind of interesting because a number of chromosomes have to come in together, and there's lots of accessory RNA that comes in at that particular point. And these chromosomes are called the acrocentric chromosomes. They're little sort of asymmetric. If you look at them, there's a little head and a sort of longer body below that. And these are chromosomes 13, 14, 15, 21, and 22. And these ends of these come close together. And all these other factors that are required to process the RNA also come together to form these structures. So initially you have a period where you have a group of them that comes together. That's the picture to the bottom right. And then there is a further coarsening process until finally you just have 1 to 2 nucleoli within the cell nucleus. So now when a cell divides, it has all of these things floating around, they begin to come together, they begin to assemble, they coarsen, until finally you just have typically one nucleolus. And all of this is happening in the background of the other stuff in the nucleus, all of the other chromosomes inside. So what we've been doing is really simulate the combination of phase separation and activity together inside this. And here are pictures of, the red regions here are regions that belong to the acrocentric chromosomes that will go into making up the nucleolus. And you can see in these simulations, the acrocentric chromosomes coming together. The time scale of the coalescence of these depends upon levels of activity. And the structure of the nucleolus itself depends upon the activity. And that's one of the results that I'll tell you about very briefly.
Professor Menon, you have 2 more minutes.
Sure, I'll be in time. So the picture to the left, the top left, is without phase separation. And the picture to the top right is with phase separation. So you can see the different chunks of the acrocentric chromosome. So these are the regions that are shown there are the parts that belong to the nucleolus. And there are little pieces of chromosome that stick out outside. So you can see that they've actually come together here. You can look at how long it takes to form the nucleolus without activity and with activity. And activity certainly speeds up the time scale that you need to get to a single coalesced sort of effective liquid droplet of the nucleolus within the rest of the nucleus. So there are some predictions that follow very specifically from work like this. It's really phase separation that helps to bring the acrocentric chromosomes together. So that's the first step towards constructing the nucleolus. But it's activity that positions and structures the nucleolus after that. You can look at the structure of the nucleolus and look at where the ATP consumption happens. It's sort of larger toward the center and lower to the outside. And that's something that I don't think has been investigated enough. Where is the consumption happening? What are the densities as you go between the inside and the outside? Many of these are deep biological questions which are being studied. But from the point of view of a biophysical understanding of where activity comes in, there isn't as yet some of these questions remain unanswered. So let me finish up and sort of, so this is work with Nirmalendu Ganai, Shivjit Sengupta, Ankit, and Tejal. Tejal has been working, the nucleolus work is ongoing work with Tejal Agarwal. All of you, most if not all of you, will remember Shurjit, who was a close friend and collaborator of many of us. The papers here are papers that reflect the evolution of our own understanding of the modeling of the nucleus, of chromosomes inside the nucleus, and the large-scale architecture of the cell nucleus. And I think, as I said in my second slide, we've begun to converge on what is really required to understand at a fundamental level the large-scale architecture of the cell nucleus, really, across different types of cells, across different types of conditions. And it's clear that the combination, that activity really is an irreducible component of understanding this biophysical problem. You need to worry about confinement because all of this happens in a highly confined region. There are sort of additional biological details that you can put in how they interact with the lamins that are really the outside, the inner surface of the nucleus as such. And these will certainly determine some level of positioning. But phase separation also seems to be an idea that is important in setting fundamental timescales for things to happen within the nucleus, for allowing parts of the nucleus to come together in a particular way that is intended for biological purposes, that has certain functional reasons for why that happens. So it's this interesting combination of biophysics, activity, statistical mechanics of non-equivalent systems, and a real biological problem that is at the center of thinking about questions such as these. And as I said, that the understanding of these, the ability to make computational and detailed models that answer very specific questions, what is the distribution of chromosome 1 or 10 or 20 or et cetera, really lies at the core of understanding the importance and the relevance of thinking about active matter in these contexts. So let me finish up. Thanks, Zahid.
Thank you, Professor Menon. Questions? There are 3 questions in the chat box. From Sriram Ramaswamy, what is the measurement that gives the local effective temperature?
So one thing that you could do is to look at local fluctuations of little labeled pieces of DNA. And from those fluctuations, infer effectively what is the strength of thermal noise or athermal noise in that region. And that has been done. And they certainly show there is evidence for inhomogeneities across the nucleus, and that you can look at the motion of small colloidal particles, of really submicron sized colloidal particles. It's a little too coarse at that scale to be able to tell this difference, but they also show differences between active and inactive regions.
No, I meant that, in the chromosome territories work in the first part.
Yeah.
Presumably, you were looking at actually experimental data to talk about the different.
That is just by a method called FISH, where you fluorescently hybridize a label to known parts of DNA, so you know what sequence is there on chromosome 21 or 18 or 19, etc. So you label it across that. And so then these are all different colors. So chromosome 18 you can fluorescently label in green, and 19 in 2, and 2 in chromosome 2 and
what is the effective temperature there?
We don't know. Because again, as I said, the only way to do that would be to look at fluctuations. But that particular experimental technique requires that you fix the nucleus. So it's dead by that time. So it's a snapshot of a dead nucleus by that time. You need to do this on a live nucleus.
Okay.
Another question in the chat box from Rajarshi Chakraborty. Effective temperature in your model is a measure of activity, but what about the directionality and persistence of the bead motion?
So that goes into questions of what level are you coarse graining at? And the idea is that if you're looking at fairly large chunks of DNA, you're looking at 1 megabase pair, that's your coarse graining length, and there you can have between 0 and 700 genes. So in reasonable measures of gene density, there's lots of stuff. There's stuff going back and forth. There's many other types of processes that are active. So the fundamental assumption here is that there is some sort of central limit that is applying. Then you can replace all of the complexities of directionality, etc., once you coarse grain enough by just an effective thermal noise or athermal noise. That's an assumption. When it would be hard to improve on that assumption, because that would require very, very specific understanding of which genes are being transcribed at what point, and what is the directionality of the DNA at that point. And I don't think biophysically you can do a better job than this at the moment.
Okay, Gautam, we'll talk later.
Sure.
I think.
Next question is from Deepak Dhar. Of course, the structure formation is interesting, but it is also interesting that at the time of cell division, the structure will be disassembled. What causes that?
That's a great question. We did think at some point that we were going to try and model even sort of the division of cells, the mitosis of cells, but that just seems to be too hard a problem. And there's a lot of protein production. There's a lot of compaction of the molecules. So the molecules become really visible under a light microscope when the cell divides at that particular point. So the whole structure of chromatin changes in ways that are not captured by this model. And so you need a different type of model to think about that. And we're not there yet. There are many other components that come in. The microtubules begin to organize, the motors come in to pull the chromosomes apart. These are much harder questions, and we have made no progress with thinking about them at all.
No, because you caused it phase separation, it seemed like inevitable that this should happen, and I'm saying the opposite also happens.
No, and you're right. So there is a way of thinking about the overlap region when cells divide and you line up the chromosomes in the middle and you begin to pull them apart. That's a sort of region where you have a combination of a nematic region in between and an overlap with a polar region. So there is some sort of subtle phase separation happening, which is not of the type that I'm describing here. But that's again something that needs to be thought about, but we have made no progress in thinking about it.
Any more questions? Okay, if not, let us thank Professor Menon once again for the wonderful talk.